This is the illustrated diagram of blood vessels. This shows capillaries, and this end they are arteries, arterioles, muscular artery and elastic artery. And this side they are veins, venules and large vein. Endothelium is always present. Other structures which make up the wall are present with respect to metabolic functions and mechanical needs. This chart shows different structures, that is endothelium, elastic tissue, smooth muscle tissue and connective tissue or fibroelastic connective tissue in different proportions. We can see endothelium, which is shown in yellow strip, is present in all the vessels, and elasticity goes on increasing from capillary towards large arteries, so they are called elastic arteries. And muscularity is more in muscular arteries or medium sized arteries and even in veins. And fibroelastic connective tissue is present almost everywhere except capillaries, because these need lesser thickness in the wall for easy diffusion. For the easy understanding and learning purpose, we divide the vessel wall into 3 different layers. This is the lumen of the vessel, diagrammatic representation. So this is innermost layer, tunica intima, and this is middle layer, tunica media, and this is outermost layer, that is tunica adventitia. Tunica intima, media and adventitia are separated by 2 blue lines here, discontinuous blue lines. These are fenestrated elastic sheets. They are known as internal and external elastic lamina. Diagrammatically, we can see them separated here. This is tunica intima, which has lining epithelium and subendothelial connective tissue. This is tunica media. It has either muscle tissue or elastic laminae. And this is tunica adventitia. Intima, media and adventitia are separated by internal and external elastic lamina. This diagram represents heart and great artery, that is aorta. In systole, pressure is exerted into the lumen of aorta. Aorta stretches because it is elastic. It is elastic artery. During diastole, the recoiling of aorta will push the blood further into the distal arteries. The elastic property of large artery is given by the structures in tunica media. This is intima with endothelium and subendothelial connective tissue, thick media and adventitia. In diagrammatic representation, tunica intima, thick media and adventitia. Thick media shows many elastic laminae which are discontinuously shown in wavy pattern, and in between them there are some smooth muscles. Tunica adventitia is thin and it has fibroelastic connective tissue with vasa vasorum. This is von Giesen's stain, where elastic laminae have been stained with brownish black color. In higher power, you can see these are the elastic laminae separated by some smooth muscles in between. Each elastic lamina with adjacent smooth muscle forms one laminar unit. In this way, there are 50 laminar units in adult aorta.
How elastic fibers are arranged in the wall of elastic artery? What is elastic lamina?
Let us imagine all these are elastic fibers. They are running single, they are branching, and the cut ends are coiled because of the elasticity. When these elastic fibers are collectively arranged to make a sheet of elastic fibers, that is elastic lamina, it will appear like this and it will show elasticity. Whole sheet will be elastic, and this is one lamina. What is fenestrated lamina? This is how elastic lamina is fenestrated. So many such fenestrated elastic laminae are present in the wall of elastic artery. When we take cross section through the fenestrated laminae, so the sheet, the elastic laminae, appears discontinued. How smooth muscles are arranged in between the laminae? If this is the vessel wall and we are constructing the media of elastic artery, so this is how elastic lamina covers, and next some smooth muscles will lie here, and then one more layer of elastic lamina. Like that, there are many elastic laminae. In muscular artery, tunica media is richly populated with smooth muscles. Strikingly, internal elastic lamina is more evidently seen in muscular artery. This is how it appears. Internal elastic lamina, which is thrown in folds. On this we find endothelial cells into the luminal surface, and all these are smooth muscle cells with very minimum number of elastic fibers in the form of lamina. These elastic laminae will decrease as we trace the artery distally in the body. The elasticity gradually decreases distally and muscularity increases as it becomes muscular artery. Tunica adventitia shows fibroelastic connective tissue. In large vein, tunica adventitia is the thickest. Tunica intima, tunica media. And tunica media has few circularly arranged smooth muscles, but in adventitia, it is longitudinally arranged fascicles of smooth muscles, which are seen in large vein. Muscular arteries branch repeatedly into smaller and smaller arteries until they reach less than 0.1 millimeter. So they are called arterioles. They usually have only 2 to 3 cell layers of smooth muscles in their wall, and the diameter of the lumen is usually of the same thickness as that of the thickness of the wall. When they contract, they can completely occlude, and they are regulatory vessels. The peripheral resistance in the vasculature is always given by arterioles. Arterioles further branch into meta-arterioles, which have very few smooth muscles in the wall, and they will branch into capillaries. Precapillary sphincters are always there from the meta-arterioles. They can open up to give the capillary circulation, or they can close. So whenever there is closure, there exists a communication between arteriole and venous end. This is thoroughfare channel. This regulation is very important in skin, erectile tissue, and in gut bed. Capillaries are of 3 types, which have continuous endothelium and basement membrane outside to them, and diffusion has to happen through the cell exocytosis or endocytosis. And certain other capillaries have fenestrations, they have holes in the endothelium. Diffusion happens with little easy manner. And few capillaries are discontinuous, which have breaks in the lining epithelium, and also they have breaks in basement membrane. These are sinusoidal capillaries. Continuous capillaries are seen in skeletal muscles, brain, and skin. Fenestrated are seen in renal glomeruli, endocrine cells, and in GIT. Sinusoidal capillaries are seen in liver and spleen.